Thanks, Kel. And I am pleased. You're going to go to the next slide, right?
It is thinking about it.
Okay.
It is.
It's a
and then we'll see,
technology
hopefully,
day.
and then it jumps too many times. Here we go.
Here we go. So thanks, Kel, and welcome everyone. And I am pleased to introduce Dr. Bernd Würsig and his cohort, who won't be here right today, but Melany Würsig. And Bernd, I am pleased to say that Bernd was my advisor for my degree work. And he actually has an interesting past. I'm going to let him talk a little bit about his stuff. But he got his degrees from Ohio State University and from Stony Brook University. Is it Stony Brook University or University of New York at Stony Brook?
It is both of those. They changed their, hello there, Kathleen and Kel. So lovely to see you and hear your voices. It is both of those. When I went there back in the early Pleistocene, in the 1970s, it was the University of New York at Stony Brook. And since then, about 20 years or so ago, they changed their name to Stony Brook University, just because I think it sounded better, and they are now a famous university.
Ah, very cool. Well, from there, Bernd went to the other side of the country, to California, did his postdoc at the University of California at Santa Cruz, and spent time after that at Moss Landing Marine Labs before he got to Texas A&M University. I'm jumping a lot of history there. And is now a Regents and University Distinguished Professor Emeritus from Texas A&M University. And in deference to the topic of your presentation, has been an advisor to more than 60 students, graduate students, and divides his time between the Southwest, the US Southwest, and New Zealand. So with that, I look forward to hearing your presentation.
Thanks so much. Thanks so much, Kathleen. It's lovely to see you again and to reminisce a little bit as well. And you know, you were one of my very first, if not the very first, PhD students actually, back in the 1990s. And of course you're now so very accomplished, and it's simply a pleasure to interact with you through the years and here in this particular forum. So thanks for that, Kathleen. That's very nice.
And thank you.
And I think what we're going to do now is to start my talk. Is that correct? And I see
so.
Only sort of a small version of it on my screen, but is it fully on the screen for everybody else?
Kathleen, what do you see?
I see all of the Adobe details and present. It's not full screen.
Do you know? Right. Is it good enough? Is there a way for me to make it larger? It fills most of my screen, but I certainly want it to look the best.
Bernd, when you go ahead.
You know, it's fine for me. It's sort of a pre-presentation type of thing on my screen, where it's maybe a third of the screen type of thing. But if you gals are happy with this, then I'm happy as well.
You might be able to go into read mode with this little to the right of your 85%. You can make it a little bigger.
Like I could go to fit to with scrolling or something like that.
Lovely.
There you go.
Yeah. It turned beautiful just now.
But now
Perfect.
It's a bit okay. Well, we'll work with it a bit. Now it's too big on my screen, but does it look all right on yours, Kathleen?
It does.
Okay, perfect. And that's the one that's recording. So that's the one that I think will be up on for everyone else.
Sounds good.
And then once again, the screen of the talk is there, and then I'm on the right-hand side or so. I can't see that, which is fine. How do you see that?
Sometimes the video is on the right margin and sometimes it's on the top. So if you can see the little video screens, it might also depend on how your screen is set up, your Zoom.
I just went to gallery view and I see Kel and I see me and then Kathleen down below. Does that sound about right?
Yep.
Okay. All right.
Perfect.
Well, then let's get started. You know, as I guess, especially as I get older, I think about the various shoulders that we lean upon, that not just I lean upon, but that we all lean upon. I do not believe that there's a particular, I actually don't believe, you don't have to agree, but I don't believe that there's a self-made woman or man. We all rely on others. We rely on our mummies and our dads, and we rely on those that have helped us through our life, our present stuff. So we do stand on the shoulders of giants. Bernard of Chartres said this. We're not quite sure when he was born, but he lived to about 1124, and I agree. I think so. And one of the big shoulders is right there. Is Mel Würsig, Melany and Caballera Würsig, with whom I've shared so much of the last 50 some odd years, which just shows you how ancient I am. And if we could go to the next slide, please. You know, one of my former grad students, Sarah Pewitz, put this together for us, and we all have the same kind of well, not the same exact backgrounds, but the same kind of background. You can go back and see whom have we relied on. In this particular case, intellectually, PhD or MD type of degree, whom have we relied on? And I see here that what she put together was that Georges Cuvier, and do when you have time, look him up, and Friedrich von Humboldt back in the 1700s were an amazingly influence to science of the time. And then Louis Agassiz, he preferred to be called Louis, I think, in the 1800s. And then he was a mentor to David Jordan and to Charles Gilbert in the US in the late 1800s and early 1900s. And they were mentors to Carl Hubbs, whom Mel and I got to meet, a lovely, lovely, wonderful, amazing scientist at Scripps in California. And then Carl Hubbs was a direct mentor to Ken Norris, who was my PhD, my postdoc advisor. And then on the other side of that scale, you have this amazing Ralph Waldo Emerson, who was a direct mentor, not direct mentor, but through several generations a direct mentor to my mentor, Charlie Walcott. And Roger Payne of Rockefeller University, and Charlie Walcott of Stony Brook University, and George Williams of Stony Brook at the time, helped to mentor me. And this is not, good folks, it's not an ego thing. It's just that we want to appreciate who came before us and not just recognize the most recent. But we want to appreciate that kind of impact that folks have had. And here we might think of Kathleen Dudzinski of the DCP, who has now expanded so much of that research on dolphin societies that really relies on some of those academic shoulders. If we could have the next slide, please. Thanks. If you click the right click on your button, I think it'll go to the next slide. Thanks for that. So Ken Norris uh, passed away several years ago, was so very instrumental in what Melanie and I did, and Roger Payne, who's still very much alive, and Charlie Walcott, and George Williams, who also passed away sadly, it had these amazing influences that I want to talk to you about today. Next slide, please. If you click the right side of that, it'll just go to the next slide. So I'm a refugee from a place in former Germany, in Southeast Silesia, and as I won't belabor you with all the trials and tribulations and fantastic stuff that we experienced in the 1950s, but then my family and I was only 8 years old, came to Fremont, Ohio in 1956, and there I am, little waif, little refugee from a displaced place after the Second World War. Next slide, please. So there's my wife. Bogota, Colombia, 1956, at exactly the same price, same price, same place in time, and here she is during her first communion, and obviously much less of a waif than I was. So I am so fortunate to have met her after she was in Bogota, Colombia, many years later. Next slide, please. The first probably important scientific influence that I had was a fellow named Hans Haas, very handsome guy. And I met him by reading several of his books and several of his papers as a marine biologist in the late 1950s, when I was still a kid, when I was only 11 or 12 years old, and in the early 1960s. And he recently passed away, that is 7 years ago, passed away. Wonderful, amazing, not just a scientist, but a purveyor, a describer of scientists. And I think he was a very important first shoulder. And I spoke mainly German back then in Ohio, USA, but Hans Haas was sort of that connection between the New World and the world that my parents and my siblings, my two brothers, and I had just left. He wrote several amazing books. One of them called, in English, Expedition into the Unknown. I don't know whether it's been translated, but I read it in German. And he had a British television show, The Adventures of Hans and Lotte, his wife, Haas. And they helped to get me started in an appreciation in landlocked Ohio of the wonders and the marvels of marine biology. If we could have the next slide, please. The second influence, slightly later than that, only slightly, a year or two or three later than that, with John Cunningham Lilly. He lived also a very good, long life. And he was sort of a new age guru, as we used to say in the 70s and 80s and maybe 90s. He wrote several books on the so-called intelligence of dolphins. And they are very bright, and they are very large-brained, and they're very wonderful, and they have amazing social capabilities, sort of separately evolved from the social capabilities of terrestrial mammals. So that's kind of fun. But John Lilly was very bright, and a lot of what he said had some truth to it, but I think, and several of us think, that he overreached a little bit. He tried to make dolphins into these super creatures that have capabilities, mental capabilities, ways of living that are absolutely amazing. And they do have ways of living, and they do have amazing social organization. But so do a whole bunch of other creatures. So do wild dogs and social social cats in the Serengeti, and so many other animals and humans. He took a lot of his thoughts during sensory deprivation chamber experiences, they're called. I have a little joke down there below, if you go just a little bit further below, that says, "Thanks for the memories." He used to lie in these sensory deprivation chambers and try to experience, also with some drugs that were taken back in the 1960s and 70s and early 80s, of obtaining knowledge that is not necessarily, and I'm not poo-pooing any extrasensory perception type of stuff, but that's not necessarily very well proved scientifically. He put brain electrodes into dolphins, and he did various other experiments on capabilities of dolphins. He had a particular experiment that was probably quite cruel to the dolphin, of keeping them in sort of a captive, and certainly a captive environment, but in a particular water environment with a trainer who interacted with the dolphin, but we're not quite sure how well that worked. In final analysis, as I think back on it, John Lilly had an amazingly strong influence on me, but I don't necessarily believe very well what he said relative to dolphin intelligence capabilities. They're bright, they're lovely, they're wonderful, they're amazing social creatures, and I think as Kathleen and as Kel has discovered over the years, and I don't mean to tell you what you're writing, but they are really cool, but they're not necessarily extra special relative to human or other capabilities. Next slide, please. Thanks for that. So my next influence, and it's coming out a little bit large in my screen, but that's okay, is number 3, Melany Ann Würsig, my dear wife. I met her when we were very young, in our early 20s, and she's stuck with me, which is amazing, and she's helped me in so very many different ways. Here she is ascending from a dive with dolphins in Hawaii quite a few years ago. She nor I look the same way anymore, but we have had a very good collaborative relationship since then. Next slide, please. Number 4 of those shoulders is Charlie Walcott. He was my direct mentor at Stony Brook University. And he came to visit us while we were doing my PhD research, Melany and I, working together on the PhD research in Argentina back in the early 1970s. Here's a picture of him in this cowboy hat in Argentina in December 1973, on Christmas Eve 1973, as a matter of fact. And on the left-hand side, he is doing one of his communing with pigeons, and he did amazing pigeon navigation work throughout his life. He was very famous for that. Anyway, he was and is a behavioral scientist of excellence. Next slide, please. He's done navigational work. He's done territorial bird communication work. Here's a very recent picture of him. He's still very much active. He's the Dean of Research at Cornell University. And on the left-hand side, he's moving an antenna in order to track pigeons that we've put little radio tags on. But it's so fun to have somebody who has influenced my career so deeply and kindly, who is still alive, still active, and without whom I wouldn't be doing the kind of research that we have been capable of doing. Charlie Walcott was an exemplary representative of the capabilities of science and of behavior and behavioral ecology and caring about conservation issues. Next slide, please. The next person, and at about the same time, just slightly after, a few months after I met Charlie Walcott, is Roger S. Payne. Here's a picture of him from 1972. He is the smartest person I've ever known. He described early aspects of bat echolocation. For his PhD, described the hearing of barn owls and how they, not using echolocation, use amazingly fine sensory capabilities to distinguish what might be a predator, a little mouse crawling along, and what might be danger. He, at the same time, in 1971, published an important paper on humpback whale song. He is the first. He and his wife, Katie Payne, and his then wife, Katie Payne, described whale song way back then. And he is the first to have described long distance, very long distances over ocean basins, communication in blue whales and in fin whales, in long distance communication. He took us on, upon Charlie Walcott's suggestion, to work in Argentina on whales, southern right whales, and on dolphins. And he's the first fellow who identified whales relative to their callosity patterns, relative to their head patterns as individuals. And he taught us how to theodolite track. I'll talk a little bit about this in a moment. How to ID dolphins. And he had indeed a bright thought every moment. And not all of the bright thoughts gelled, but he had a bright thought every moment, and he still does today. He's also in his 80s now in this year in 2020. Next slide, please. Here's a fun picture of Roger Payne communing with a young butterball pup, we call it, a southern elephant seal. And he wrote, this reminds me that he wrote an amazing book that if you're interested at all in the science of cetacean, of whale and dolphin behavior, you should read. It's called Among Whales, 1995. Beautiful book. He helped to energize the anti-whaling movement in the Whaling Commission aspects, and he helped to get the moratorium of 1986 of no more whaling into the international regulations and ways of proceeding. He's now the head of an organization that he started called Ocean Alliance Incorporated that has done a heck of a lot of good on ocean situations. Next slide, please. So because of Charlie Walcott and because of Roger Payne, we made it to Patagonia, Argentina. Absolutely beautiful. If you get a chance to travel there as a scientist or as a tourist, please do so. It's a beautiful, beautiful place. These are some of the shots of where we lived, and I won't go into detail. Next slide, please. Oh my goodness, Patagonian life on land with the lowland guanaco on the left middle there, with the Pecho Colorado, the red-breasted meadowlark on the right-hand side, with the fox down below, and the little queese and the rattleless rattlesnakes. Is absolutely a gorgeous environment to learn about nature with right whales and bottlenose dolphins and dusky dolphins right offshore. Next slide, please. Here are some of those animals listed. The guanaco, that lowland llama type. The Pecho Colorado, the red-breasted meadowlark. The hairy armadillo, called peludo, which means the hairy one in Spanish. The lesser rhea, the Patagonian gray fox. The amazing Patagonian beautiful tarantula, huge tarantula. The red-backed race of the variable hawk. The tucu-tucu on the left bottom that communicates through channels of many hundreds of meters with its conspecifics about danger and about prey. The next slide, please. So we spent time there. We learned a lot about dolphins and whales while we were working for and with Roger and Katie Payne. And the first paper that we wrote is actually a paper that Melany Würsig, my wife, is a senior author of, and it's called the occurrence, the behavior, and the general situation of the Burmeister's porpoise, Fission spinipinus, in Chubut, Argentina. And it's not an amazing paper because we didn't know terribly much about the animals, with Jorge Mermoz, by the way, a wonderful scientist from Argentina. And it was our very first paper back in 1977. Next slide, please. But we learned, especially from Roger and Katie, and we then sort of developed it a bit more, of course, and became well known for it. But we learned that you could identify dolphins mainly by the trailing edges of their dorsal fins. And it was an exciting time in the 1970s. We had learned, others had learned, that elephant ears and leopard spots and chimp faces and killer whale saddle patches and right whale callosity could all be used to identify particular individuals. And as soon as you can identify individuals, good folks, and I think you know this, you can start saying something about who's with whom, who's spending time socializing, maybe sexualizing, having young, with whom. And as soon as you can do that, you can start describing aspects of social biology of creatures. And indeed, Roger Payne and Katie Payne and another colleague who was doing similar work all on her own in Texas, Susie Shane, helped us get that organized. And today, there are, well, as you know, the DCP is so important for that. There are hundreds and actually maybe a few thousand projects using identification of individuals in order to look at social strategies. Next slide, please. So we wrote a paper about that, the photographic determination of group size, composition, and stability of coastal porpoises by Mel and me. And yeah, we did it, we proved it, we showed it. But the point is, we weren't the only ones. It was those shoulders that we relied on. It was Charlie Walcott and Roger Payne and others that we relied on in order to get to the point where photographic recognition could actually be used, be accepted, be something that would go into the future. Next slide, please. We used onerous methods of comparing hundreds and thousands of photographs in order to see who's who. These days, it's getting much more easy to do that by individual identification. We started a particular program called FinScan, that is now, I don't think it's actually been surpassed. It's now not the latest bit anymore. But with FinScan, you can do the outline markings of particular animals of the dorsal fin, and then get a bunch of choices of which might be closest, and then finally come to a conclusion. Ah, this is that one. This is that one. This is number 1, 2, 3, 4, 5. So that is becoming ever more sophisticated now, with individual recognition signatures is becoming very easy. But believe me, it wasn't 20 or 30 years ago. It relied on people spending hundreds of hours, thousands of hours, in order to see whether from a population of, let's say, 300 individuals, this particular animal spent time with this particular animal, etc. Next slide, please. We learned photo identification recognition, and that was very valuable. But we also learned, and it was while working with Roger and Katie Payne, we also learned the use, the amazingly powerful use of surveyors' transits, or they're called theodolites, is a sort of technical term. If you're on shore, and you're looking at animals that are close to shore, within sort of 7 miles, 10 kilometers, that kind of thing, then you can use that theodolite that Mel's looking at right now. That's a very primitive one called DKM current theodolite, a Swiss theodolite. You can use that if you know your exact station height on shore. Then you can use that in order to get exact positions of an animal that's traveling offshore. And that means that as you get more and more position, you can get speeds of travel, you can get distances of water that they're in, you can get orientations and reorientation information that is very sophisticated relative to as long as animals move offshore. The way Mel saw, and she became the world's expert at the time in the 1970s, the way she saw the animals was upside down, because of current DKM1 theodolite did not have a correction factor for upside down. So everything was upside down and moving left to right instead of the appropriate right to left. And she and I still see the world that way sometimes, upside down and left to right instead of right to left, or vice versa. Several students of mine, Glenn Gailey and Joel Ortega Ortiz, in the early 2000s, they're also wonderful shoulders, developed a technique, a computerized program called Pythagoras, in order to efficiently analyze theodolite tracking. Next slide, please. And there you have to have your station and the station name. You have to know your exact station height in meters, and the eye height, that is the height of your theodolite. You have to know, it doesn't say that here, but you have to know your height of eye relative to the particular changes of the height of you relative to the height of the sea, compared to the particular tidal situation at the time. And so you have to know your exact station height, your exact location. You have to know a particular 0 point left to right, and then you want to gauge, while you're looking through the theodolite power scope, you have to gauge your behaviors, and you have to say when you're exactly fixed on a particular dolphin or a particular whale, in order to get a reading of its position out there 8 kilometers away, in order to get several readings, in order to get speeds and travels and orientations and reorientations. Kind of cool. It's a neat system. So first, photo identification. Second, we learned how to properly structure and then analyze theodolite tracking information. Next slide. So our next shoulder is Kenneth S. Norris, and he also passed away quite a bit, over 20 years ago. He's the fellow who discovered how dolphins hear by discriminating echolocation clicks left and right in the sides of their heads. He described how the click whale structure of sperm whales allows them to assess another sperm whale, how old they are, how big they are. He's the person who taught us about sensory integration of animals and started to teach us about dolphinid societies. Most of his work, not all of his work, but most of his work on wild dolphins was in Hawaii, on Hawaiian spinner dolphins. Next slide, please. I think he's the first one who really taught us that the dolphins are bright, for sure, have amazing social organization, have amazing capabilities, as John Cunningham Lilly had said earlier, but that they also have amazing problems. They can go hungry. They need to find food. They need to dive down in order to get their food. They need to work for it. They need to contain it. They need to corral it. They need to socially organize in order to get enough food to survive. And they have dangers. They have large sharks, white sharks and other sharks. They have killer whales, for heaven's sake, are the bigger cousins, their bigger cousins, the largest of the delphinids, killer whales. They have bad weather to deal with, and they have humans to deal with, noises and pollution and accidental netting and other problems. Next slide, please. Dolphins, and I guess we sort of learned that with Roger Payne and Katie Payne and in Argentina, but I think Ken Norris helped to formalize that with his shoulder. Dolphins are highly social creatures. Whether they live in society of dozens or in thousands, a lone dolphin is a rare dolphin. And I think Kathleen and Kel caught into that and did progress that information so much better with bottlenose dolphins and with Atlantic spotted dolphins and with others, that they are highly social creatures. And they live long lives. They have long-term associations, relationships that are very complex. And we started appreciating that back in the 1970s. And again, that appreciation and the knowledge for it came to the fore in the 1990s and the 2000s. Next slide, please. And their societies tend to be of a fission-fusion type, where there are recognition of at least some long-term partners. There are recognitions of individuals, like we recognize them by their dorsal fins, but they recognize themselves and each other by amazingly more sophisticated cues. There's long-term memory of whom you've dealt with. Probably preferential partner choices. We have some good evidence for that in some societies. And overall, on sort of a larger scale, you can think of the concept of cost-benefit choices, of what are we as humans in our societies, and they as animals in their societies doing relative to those choices. We call it the biological markets paradigm. How much will it cost me? How is the best way to behave? And is it worth it to behave this way or that way? If I'm going to be a dork, if I'm going to be a bad behavior, is that really the best way to go in the long term? Or do I play a better game and be kinder and work with those colleagues, those others that I'm living with, in order to myself have a better relationship and do better in life? If I offer enough, what am I getting in return? I don't like putting it into exactly those terms, but I think you might know what I'm talking about. That we're all basically selfish human beings and selfish animals. But if we look long-term enough at relationships, then maybe we can do better. We can relate better to those others, and that will eventually help ourselves better also. Next slide, please. So we worked in Patagonia with dusky dolphins, bottlenose dolphins, and I didn't tell you terribly much about the details of that, but we learned about societies and society structures, and we certainly learned that there were lots of shoulders that helped us along the way. And then we worked in Hawaii with Ken Norris on Hawaiian spinner dolphin, and we learned more about societies. And then Mel and I were very fortunate to go to study dusky dolphins, not only in the shallow waters of Argentina, Patagonia, Argentina, South America, but in deep waters of a new place, a different place, a world apart, about 180 degrees longitude apart of the New Zealand South Island. And we found there that the animals are foraging very differently from Argentina. They're now no longer in shallow water, they're in deep water. And in deep water of particular areas, they can only find good food that is in those deep waters that rises towards the surface at night. It's called an aspect of the deep scattering layer, the DSL. Look that up if you'd like to. And the food is way down below, 800 yards or 800 meters, almost a kilometer or so below during the daytime, but it rises towards the surface at night. And these animals then have the kitchen open only during the nighttime. They can only feed on those animals that have risen towards the surface, where indeed they can access it. They're divers, they're dolphins, but they can only dive down to 50, 100, 140 meters or so. And to go deeper than that is impossible for them. So they have to wait for food to come up at night. Next slide, please. And it's kind of fun. This is sort of a summary slide of what was up on the top one that I didn't describe properly. The yellow here is the rising food. The blue is the, excuse me, I'm got it totally wrong. The yellow is the dolphins and the depths that they go to. On the left-hand side, you've got meters, 25, 50 meters, etc. On the bottom, on the x-axis, you have time of evening and of night and of early morning. So it goes from 500, from 1700, that's 5 o'clock in the evening, to 5 o'clock in the morning. So those 12 hours are represented here. From 5 in the evening, that's 1700, to 5 o'clock in the morning. And you have the deep scattering layer rising, that blue stuff there. That's the fish, the myctophids, the lanternfishes, the stuff that dolphins feed on, rising towards the surface. And it's nearest to the surface at about 60 meters or so depth. That's about, 60 times 3, that's about 60, 120, about 170, 180 feet deep in there. And that's when it is easiest for the dusky dolphins to feed on that fish, on those fish types. And that's around midnight. And then it gets harder and harder for them to go down in order to feed. Dusky dolphins seem to not feed deeper than about 130 meters. They can dive a bit deeper than that, but it's just not efficient for them to do that. So they can only feed during the deep scattering layer rising of the food during the nighttime. Next slide, please. And that was cool. That was really a cool thought. Now remember that we're identifying individuals at the same time. And we're looking at these New Zealand dusky dolphins in deep water near what's called the Kaikoura Canyon, where they feed at night on deep scattering layer related organisms. And then we recognize particular individuals. There's 2 fingers on the left, and there's a Nike swoosh on the right, and that sort of really ugly one, my graduate students named Bernd. For some reason, they always name the ugliest dolphin after me. And we found out that those dolphins, those recognizable animals that feed on the deep scattering layer at night in deep water, Some of them migrate to shallow waters in an area north of there called the Marlborough Sounds, and they change their day-night regime. It's shallow water, so there's no deep scattering layer. So they feed on schooling fishes in daytime, and they associate differently, and they work differently, they behave differently relative to the food availability patterns. Nighttime in deep water and daytime in shallow water and other areas. It's kind of cool. So we did discover that, and the only reason we discovered that is because of people like Roger and Katie Payne and Ken Norris and others. Next slide, please. Oh yeah, there it is. That's a highlight, just to remind me to tell you that the ugliest dolphin is always named after me. Next slide. So recently we've been learning something new about dusky dolphins, and it turns out about quite a few other dolphins. And I think Kathleen and Kel already know quite a bit of this and learned quite a bit of this about their dolphins, bottlenose dolphins, Atlantic spot dolphins, and others in their societies in more tropical waters. All of us learned at about the same time that there's a type of scramble competition by males towards females. It's what we call a multi-mate, or polygynandrous is the appropriate term, society, where both males and females mate with several other females for the females, and several for the males, and several other males for the females. So it is multi-mate. In human terms, it's often called promiscuity. I don't like the term because promiscuity implies the absence of mate choice, whereas I think they're making important mate choices, especially the females are making important mate choices. They only have one youngster every 3 to 4 years or so, or 5 years. They need to make very, very sure that the male that they're mating with, the males that they're mating with, are appropriate, are good males, however they choose that. And we are starting to learn a little bit about what they're choosing. But I think it is not promiscuity, it is organized multi-mate type of stuff. And there is scramble competition of males trying to mate. These are all males leaping after a particular female who is ready to mate, but she's making it difficult for them. She is choosing a particular male or a particular male over others in order to mate with. And while the males are scrambling, the females are choosing. And I think we'll learn more about this as we go on, but it's an important point, and we're starting to learn about that in dusky dolphins, in deep waters of New Zealand, and shallow waters of Argentina, and in several other species. Next slide, please. So, yep, shoulders. And we're still learning so much about who has helped us. And we're learning quite a bit about social strategies. And we're also learning that we don't know all that much about the finalities of social strategies. But we do have amazing marine mammal concerns. And I want to end with this. We have on the left-hand side there, monk seals. That would be Hawaiian monk seals. We're not doing very well at all, although better this year and the last 2 years than before. We have sea otters in various populations. We have polar bears that are suffering due to overall large-scale climate change problems and less ice than there used to be. We have western gray whales off Kamchatka and other parts of Russia that are not doing well. We have the little, in the middle bottom there, the little vaquita, the Gulf of California harbor porpoise. It's hanging only on by a string because it's being caught in set nets for illegal catching of large sea bass in the Sea of Cortez south of us in the US. We have various types of seals and fur seals. We have the baiji on the bottom left-hand corner, the Chinese river dolphin that is extinct now. It has gone extinct. And and to the left of it, the vaquita may follow, simply due to overfishing and what's often called human greed, but it's not one particular person or company's fault. It is the overall degradation of the environment by humans. It's a big problem. Next slide, please. So we have bunches of environmental concerns, including fisheries competition and entanglement and physical pollution and ship strikes and sound pollution and habitat loss and degradation from too much tourism, often with large ships, and competition with other major industries. I think the biggie is global climate heating and ocean acidification. I think that's the real biggie that we have to confront and hopefully are confronted internationally and hopefully eventually nationally in the US. Next slide, please. Whenever you point out problems, you want to think about potential solutions. You want to think about ways to help. And a major situation might be that red marked one, government and major non-government organization buy-in in order to help change ways of operating relative to fisheries, relative to pollution, relative to sound pollution, relative to environmental degradation. And a lot of other possibilities come with that. But I think the overall government and major non-governmental organization buy-in is very important. Next slide, please. Thanks for that. I like the idea of being able to look at problems and being able to help solve them by being flexible in management strategies. We keep on hearing, or we've often kept on hearing, that we must get more data. Well, we don't need more data in order to tell us that vaquita, the Gulf of California harper porpoise, is in terrible trouble. We can resist that scientific call for more information. We can act now. We need to act now in order to, in that particular case, stop illegal fishing on the giant sea bass, totuaba, that's incidentally capturing the little vaquita. And we need to do that now and very efficiently. We don't need more scientific data. I think overall it comes to a point of societies, and you can help with this, good folks, of societies honoring nature as much as we honor ourselves. That is, when we talk about, oh, we need to have an increase of 3% of GPS of the general increase in monetary capability for this year or the next year. We need to have that kind of thing. Well, maybe we don't need to have that kind of thing. We don't need to buy another refrigerator. We can wait for another 5 years. We need to think about honoring nature as much as we want to advance ourselves. And that's a toughie. That's toughie, because everybody needs to work and live and have a decent life at it. But I think as we, and as some societies have, human societies have gone to the point of honoring nature as much as ourselves, we are slowly, maybe possibly advancing. Next slide, please. There are successes, for heaven's sake. Yep, vaquita, the Gulf of California harper porpoise, is hanging on by a thread and might die out, darn it. And the Chinese Yangtze River dolphin has gone extinct, and that's really, really sad. But eastern gray whales in general, are doing much better than they were 15, 20 years ago. Those dusky dolphins that are now thriving off both shores, Atlantic and Pacific of the South Americas, and thriving in New Zealand are doing better now, are indeed better thriving than they were 30 years ago. There are still problems, but off New Zealand, for example, where Mel and I do a lot of work, there were major PCB and other chemical pollution problems that were killing thousands, unknown thousands of dusky dolphins. And that's been cleaned up. They are no longer doing that. We are in a better situation. So we need to think about the positive aspects as well. Next slide, please. And instead of thinking just about the next job opportunities and how well we're doing economically, which we need to think about, I know, we want to think about the future of us as humans, and of us as humans is intricately tied up with nature. Do we want, indeed, the Western gray whale, which is quite endangered, unlike the Eastern one, off Russia, the little vaquita, the Gulf of California harbor porpoises, the pods of killer whales or orcas that are not doing well at all because we've overfished salmon, and the Eastern Taiwan Strait Indo-Pacific humpback dolphin that's not doing well at all because of amazing industrial pollution off Taiwan that I haven't described. But we want to make sure that 100 years from now, these animals will do better, and that we will enhance their environments and their ways of living in order for them to do better. Right now, the Tethys Sea, I like to call it, but it's really the Mediterranean Sea, or what's left of the ancient Tethys Sea, is a cesspool. It's been overfished. It's been amazingly degraded by sound and by chemicals from various countries. Nobody's particular fault, but various countries. And we want to make sure that we can gain enough political pressure and environmental conservation understanding and relief in order to have the Tethys Sea, the Mediterranean Sea, become alive with food, and therefore with dolphins and whales again. Next slide, please. This is a bit shifting. About 30 years ago, it was guys, it was men, it was male human beings who were in charge of lots of things, politics and science. And since then, we've done a bit better. We now have, in our breed of science, in marine biology, and I think in marine mammal biology, we're doing better. We have maybe close to 50%, and I hope over 50% actually, of females involved as well, female humans, of women involved in science. And why the heck not, for heaven's sake? Why would you exclude societally, purposefully, or just because of the way the winds have blown, 50% of a group of humans? And we're doing better in that gender situation. We're not doing so well in the race human situation. I kind of dream of, in my old age, of the next generation of Star Trek. I don't know how many of you people realize that these are members of the flight crew, of the direct flight crew of the second generation, the next generation of Star Trek people. But I kind of dream of it not just being men and not just being women, but being black and brown people and others from all kinds of cultures and backgrounds. And why the heck not? Why be exclusionary there? So we have to work, and I think we have to work very actively, no matter who we are, in order to make sure that people of different races and backgrounds are properly represented and represent us, represent science and education and conservation efforts properly throughout. Next slide, please. I see positives. I see positive aspects. I see Kathleen there on the top left. Dr. Duchinsky. Yes, yes. I see Lisa Balance to her right. I see Mel in the middle there, Melany Würsig, my wife. I see a bunch of other people who may not be as well known, but have been wonderful students of mine, who are now graduate students in research and activities, and others. I see Dara Orbach down on the bottom left, who is now an assistant professor at Texas A&M University Corpus Christi. And I see Kel. I see Kel right there. Yes, yes, yes. Thank you for highlighting her. As the present and the amazing future that we have. These are just a few of the associates that I've worked with in the last few years. And I want to emphasize to us as we leave here that it is important to not just know a bit about science and about dolphins and about dolphin societies and about conservation efforts, but to know and to realize and to help further realize that we as societies from all possible genders and all possible races and creeds of humans need to help. Thanks for that.
Thank you, Bernd. I had to unmute and pull my video up, and I don't know how to do the clap with Zoom, so I'll just do it just as a clap right there.
Sorry to get so misty-eyed here, but it's just me. I'm getting that way in my old age, and I think it's important.
That's okay. I thought it was great. I'm going to defer to Kel on the last few wrap-up slides, and then I do have a question for you that came up when you were speaking, if you don't mind.
Not at all.
Okay. Lovely. So just our usual reminders to folks that if you missed a webinar or you have a friend who wants to view this recording, you can find those on our website, dolphincommunicationproject.org. Just head to the education menu and there you'll find the webinar page. And in the first or second slot is also the schedule for the upcoming 2 or 3 sessions at a time. And then we also have our podcast, the Dolphin Pod. So if you haven't already listened to that, you can access it wherever you get your podcasts. Our next deep dive is next Thursday, the 12th. I'm losing track of the months and the weeks now, but we will be joined by a PhD candidate who uses passive acoustic monitoring to better understand how bottlenose dolphins use Florida estuaries and things. And then we have a really fun dolphin lesson coming up, a little deviation, and we will be talking with our friend Suna about being a zookeeper. So something a little more light-hearted for the younger and the young at heart. And of course, DCP is a nonprofit, so we would be remiss if we didn't remind you the ways you can support us. You can adopt a wild dolphin from our Bimini study site. You can become a member, get a t-shirt. When COVID is over, you can come join us in the field, all sorts of ways. And of course, just learn, use our website, learn, gain information, share it with your friends, and keep in touch with us on social media and by email. And with that, we will dip into questions.
So the question that came up was, and I had to write it down while we were looking so that I didn't lose it. How did you know which Dusky dolphins were doing the diving? Did you tag them? How did you know how deep they were going and for that study when you showed the graph?
Yeah, no, that's an excellent question, Kathleen. Yeah, we don't have particular individuality there. The way that a study worked is very happy, is working with a wonderful researcher, Kelly Benoit Bird, who worked with Whitlow Owl, her major advisor, who just recently passed away. from Hawaii, University of Hawaii, in order to track dolphins with sophisticated fish finding radar, fish finding sonar, excuse me, and she and her advisor Whitlow Au devised a technique to use 3 transducers that work together. One of them at relatively low frequency, just above our hearing at about 35 to 40 kilohertz. One at a medium frequency, which to us is way, way above our hearing at about 85 to 100 kilohertz. And one of them at higher frequencies, which is indeed totally beyond our hearing as well, at over 120 kilohertz. And what happens is when you use these fish finding sonars that project signals down to different depths, the higher frequencies can't quite reach the total higher depths, but they can give, because higher frequencies make smaller wavelengths, they can give higher resolution, higher sort of acuity from an acoustic perspective. And the lower frequencies can reach down lower, but don't have quite the same what we might call in visual terms, that would be sort of the pixel size of a picture. They don't have quite that resolution. But when you use them in combination, that sophisticated sonar capability in combination of the three units, then you can actually track dolphins from the surface going down. They're in the sonar beam, but at very, very high frequency. Not that much intensity, so it doesn't seem to be bothering them, but they're in that sonar beam and they're very used to fish finding sonar from lots of fishing vessels going around. And you can track them by the reflections, especially of the air pockets within the dolphins. First, the lung volume, air pockets. And then as the dolphins go down, the lung volume decreases because the amount of volume of air decreases as they go down to different depths. But you still get the air reflecting from their various aerial passages. You get a signature of the dolphins going down from 0 or 10 meters depth down to 130, 140 meters depth. And you can even tell the front and the back of the dolphin by characteristics of that sonar coming back. So it's not individual identification in this case. It's just tracking animals going down and tracking the prey at the same time because of their air bladders. So it's kind of cool stuff. It's way beyond what I'd be able to analyze, but Kelly and Whit and others have been able to analyze that very well and have been able to get us sophisticated new information. Does that sort of answer your question?
I believe it does. It does for me. I believe it does for I hope for the person who asked. So very cool.
But the point is, it's fish finding sonar.
Yeah.
That's getting more and more sophisticated. Mm-hmm. And these marine biologists have made it quite sophisticated. But that's really what it is. It's finding out where the fish are and where the dolphins are attacking it, because dolphins have air pockets within their bodies.
Very cool.
That reflect, that are at different density.
Yeah. Very cool. Very cool. But do you have a favorite? I know you have a favorite species, or at least over the years, I think that you prefer the Dusky dolphins for dolphins. But do you have a favorite field study site that you've been to?
Both of those are good questions because, as you know, Kathleen, all animals are so cool. And so sure, I've worked a lot with Dusky dolphins, but there are lots of other really cool animals, and certainly cool marine mammals. And Mel and I have worked a lot in Patagonia, Argentina, and a lot in the Amazon with river dolphins, and I think so fondly of the kind of work I did with you back in the Atlantic with Atlantic spotted dolphins years ago. Those are really fond memories. But sure, dusky dolphins of New Zealand in both the nearshore, the shallow water, and the nearshore deep water environments have been very instrumental in helping us to learn about societies and about foraging strategies. I suppose in final analysis, it is New Zealand, Kaikoura, beautiful environment with lovely dolphins and sperm whales that are very most attractive. But you know, that environment is a relatively, it didn't used to be, but it is now a relatively pristine one. The dolphins are doing well overall, and that's good. We need to learn what makes environments good and what helps the animals and plants in the environments to survive well. But it's not necessarily the most important research. The most important research is probably on critters like in the Arctic that's surviving so very badly with climate change. So that I'd love to know more about problems and the ways to help solve them of polar bears, and which are wonderful marine creatures, and of narwhals and beluga, and the kind of problems that they face. So your question is good on a sort of a personal level of what do you prefer. But I'd like to see, I guess, us do more than I prefer and actually work with creatures and ecosystems that need desperate help.
Well, that was going to be my follow-up question, was what would you, if you could do anything in terms of a research question or a species or a location, what would you do? And I guess that's something you can answer personally, or you can answer as you just did.
Yeah, no, I know. I've written several papers on that recently. But the summary is that I haven't done very well. I've done well in studying animals and learning about science, but I haven't done very well, and I mean that. It's not sort of an expression of humbleness. I haven't done very well in learning about animals relative to conservation and conservation needs and helping us further those. If I had to do it over again, your question is a very good one. I'd probably work with, it's fine to work with animals that are doing well, and that's good. You want to know what helps to make them do well. But I'd probably work, Mel and I worked with Baiji, the Chinese river dolphins, right before they became extinct. And we tried to help, but we were unsuccessful. They went extinct. We obviously didn't do some of that right, or sufficiently enough. If I had to do it over again, I think I'd concentrate more on really beleaguered situations and populations, like in Malaysia, with several dolphins and a few riverine ones, and in India and the Arctic situation I just mentioned. There are some real problems out there, and the more we who pretend to know something about social ecology and societies can help with the more beleaguered ones and make recommendations at higher levels and get local people maybe to change ways, the better off we will be. So, yeah, I'm not apologizing. I'm not regretting what I've done in my life, but I think to do it over again, one would be more proactively a conservationist instead of just more science, more knowledge.
Well, I think we won't take any more questions. I think I've monopolized your time on that. But I would say as a set of shoulders that I have used in my career, I think that you did encourage your students to consider conservation and consider education and not just the science, because it's about sharing what we know, not just with the academic or scientific communities, but with the broader audience as well. And I think most of your students, I haven't met all of them, but most of your students have gotten that message, if I could speak collectively for them. And thank you.
That's great. Thanks, Kathleen.
So I don't know if you have anything else you'd like to say, or if Kel would.
Yeah, I think
we're...
this is, unless you have something, last words you wanted to conclude with, I think that's a nice moment to sort of leave off that inspiration, perhaps, for the next generation to keep conservation in mind. And I think it applies not only to scientists. I think everyone can approach their day-to-day life as a conservationist in a more meaningful way.
You said that very well, Kel. Yeah, and my only addition is to thank you too for giving me the opportunity. And Mel is right here in spirit. She's just quite shy, as you know, and she would rather be behind the scenes and really do good things, and then later on chastise me for not mentioning this or that.
Those are the good conversations after, right?
Excellent. Well, thank you so much for joining us today and sharing all of that. And thank you to everyone who joined live. And thank you to everyone who's listening to this as a recording. We hope to see you all again soon.